Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing ICMR BRET exam question paper. I am discussing the microbiology question paper. Cross-disciplinary I have already discussed, and if you this is of the year 2024. If you want previous year question papers of ICMR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, see for ICMR, and you will find them there. If you are new to my channel, please do like, share and subscribe. Then coming to the first question, what is trans-ovarial transmission? So for example, I am taking the case of an infected female mosquito. Okay. Now the mosquito is being infected by some or it is a carrier of some pathogen such as virus, bacteria or any other parasite. Now this infected female mosquito, it will pass this pathogen directly to its offsprings through its eggs. Okay. So and the larva which comes out of this infected eggs is also infected, and once this larva grows to an adult, it is capable of transmitting the disease. That is called as trans-ovarial transmission. So here if you see the options, you can see that transmission of pathogen from mosquito to human through ovary. Okay, so the pathogen, one mosquito, the female mosquito is infected by the pathogen. It will transmit this pathogen to its offsprings through the eggs. The larva becomes infected. The larva grows into an again infected offspring or adult, and it will go and bite a new human being. It will be able to transmit the disease. Okay. That is called as trans-ovarial transmission. So answer here is option 2. Then what is EIR? EIR is entomological inoculation rate. It is a metric. Metric means just like you have centimeter and meter for measuring the distance. So metric for distance is centimeter and meter. So just the same way, here it is entomological inoculation rate. It is a metric and it is used to measure the intensity of malaria transmission in a particular area. Okay, that is entomological inoculation rate. Then next one, which of the following is vector of Chagas disease? It is Rhodinius prolixus. So answer is option 1. Then example of soft tick. Now there are two types. One is soft tick and the other is hard tick. Now soft ticks, they don't have the hard outer shield or the black plate, also known as the scutum. Whereas the hard tick, it is having this hard plate or known as the scutum. Now soft tick, they appear to be like more like a wrinkled looking body. They don't have this hard outer covering. Okay, just like in case of hard tick. Now soft ticks are known to cause the disease like that is tick-borne relapsing fever. And the soft tick that transmit this tick-borne relapsing fever, it belongs to the genus Ornithodorus. So here the example will be Ornithodorus movata. Then next one, which of the following disease is caused by sandfly? It is leishmaniasis, also known as Kala-Azar, and Kala-Azar is visceral leishmaniasis. So the disease caused by sandfly is option 3. Babiosis, it is transmitted by black-legged tick or Ixodes scapularis. Then you have got yellow fever and Zika, which are spread by the bite of infected mosquito, especially belonging to the Aedes species. Okay. Then next one, what kind of mouth part are present in housefly? Housefly is having a sponging and lapping type of mouth parts, and these mouth parts, they are modified for sucking up liquids or semi-solid food. Then next one, what is extrinsic incubation period in dengue? So extrinsic incubation period in dengue is 8 to 12 days. Now what is that? So if you see option 2, they have mentioned the duration when mosquito takes infected blood and the pathogen appears in the salivary gland of mosquito. So what the statement is saying is that dengue, you know, it is Aedes aegypti, which is the one responsible for the spread. So the female Aedes mosquito, it will bite a person who is infected with the dengue virus. So the virus, it will enter into the mouth of the mosquito and it will spread to the mosquito's salivary gland. And what will happen after 8 to 12 days of this replication and everything of the virus in the mosquito salivary gland, it will be capable of the mosquito will be capable of transmitting the virus to a new person when it go and bites the new person. So this is called as extrinsic incubation period and it is 8 to 12 days. That is the duration when mosquito takes the infected blood and the pathogen appears in the salivary gland of mosquito. Okay. So the pathogen has to replicate in the mouth of the mosquito, enter its salivary gland and get ready to be transmitted to the next person. That is 8 to 12 days. That is called as extrinsic incubation period. Then what are cecropins? Sorry, they are antimicrobial peptides. Now they were first isolated from the hemolymph of Hylophora cecropia. Okay, they are a part of the innate immune system of the insect. And what are they capable of doing? They are capable of lysing bacterial cell membranes, cause leaky membranes and even inhibit proline uptake by bacteria. That is what cecropins do. So they are constitute or they are one of the constituent of innate immune system of insect. Then given below two statements, you have to say which statement are correct, whether both are correct or not, or one is correct, other is wrong. Okay, so the mosquito is a day biter. Okay, the statements are about Aedes mosquito. Okay, Aedes mosquito. So the mosquito is a day biter. Yes. Breeds in freshwater container. Yes. And spreads dengue, Zika and malaria. No, not malaria. Malaria is by Anopheles mosquito. So that statement is wrong. So when you are reading the statement, read each and every word carefully. Then the mosquito is a day biter, likes to bite human, of course, and spread dengue, Zika and chikungunya. Yes. So first statement is wrong. That makes option 4 as the right answer. Statement 1 is false, but statement 2 is true. So the answer is option 4. Then horn fly. Horn fly is a parasite of horn fly is Hematobia irritans and it is a parasite of cattle. Then what is referred to as MHD in malaria? It is Migration Health Division. Now migration, you know, you are going from one place to another. Now migration can result in the spread of malaria into new areas, or it can reintroduce malaria into areas where it was previously eliminated. Means suppose I am infected with malaria and I am going from South India to North India. So what will happen is I am going to a region in North India which is not having malaria there. Suppose I am just telling hypothetical. Okay, so I am going to the place where it is not having malaria, or It is to an area where previously malaria was there, but the health workers and other workers have worked hard in such a way that they have eliminated malaria from that region itself. So I am going to such a place and I am infected with malaria. So what will happen is I will be reintroducing malaria into a place which is already eliminated of malaria. Or if the place, as I told you, it is a hypothetical place which is not at all having any malaria mosquitoes there, I am introducing a new one there. I am introducing the disease. Okay. So what this migration health division will do is they will address such kind of unique risk and challenges of malaria among the migrants, and they will suggest prevention strategies to the migrant populations, and they also will work in coordination with the government and WHO, and they will create awareness among the health workers. They will train the health workers as well as they will build infrastructure for diagnosis and treatment of malaria. That is what is referred to as MHD in malaria. Then next one, again two statements are given. You have to say whether both are correct, both are false, or one is correct or other is wrong. Okay, male mosquito take blood but cannot transmit disease. That is not a correct statement. Male mosquitoes do not feed blood. They consume nectar or plant juices for their energy sources. Okay, and so they cannot transmit the disease. Now what does statement 2 say? It says male mosquito cannot take blood. Okay, hence do not transmit disease. That is correct. So statement 1 is false and statement 2 is true. That makes option 4 as the right answer. Now next one, what defines best for ACL2 in vector biology research studies? ACL2 is arthropod containment level 2. Now what is that arthropod containment level? Now it is usually practiced when you are working with either exotic species of arthropods or indigenous arthropods that are infected with biosafety level 2 type of diseases. Means suppose you get a new arthropod from a foreign place and you are bringing it into your laboratory to see to study the various features of this arthropod. You have to be careful because you have to follow arthropod containment level 2. It may be some arthropod which is causing a disease in humans or animals. So the laboratory personnel who is handling this arthropod, he or she is having more chances of getting the arthropod disease. So that person has to be careful when working with such kind of arthropods. Or suppose you are, now this is not an exotic one, you find that arthropod in your surroundings itself, such as you take the case of a mosquito, you and it has, this mosquito has resulted in a disease outbreak in your region, and you are the one who is responsible for studying this arthropod biology and everything, and other research techniques have to be applied on this particular mosquitoes to study the cause of this outbreak of the disease spread by the mosquitoes. So you have to be careful and you have to follow arthropod containment level 2 because you will be working with live mosquitoes, not with killed or cold immobilized mosquitoes. Okay. So live mosquitoes means chances are there that you are surely going to get the bite of that particular mosquito. Okay. So you have to follow arthropod containment level 2. Now next one, which of the following is component of circulatory system of insect? It is the hemolymph. Whereas the other component of circulatory system of insect is dorsal vessel, and dorsal vessel, it is divided into two parts, that is aorta and the heart. Now thorax and abdomen, they are segments of the insect body and not components of the circulatory system. Fat body system, it is for energy storage. So the answer is option 3. Now, which of the following is responsible for cellular immunity in insect? So it is by again by hemocyte or the hemolymph. So answer is option 4. Then what is the best feed source for mosquito larva? Means what does mosquito larva likes to feed on? Or what is its diet? So it is mainly algae and some bacteria. So here the option is option 3, that is microbes. Then which insect of the following serve as best model for genetic studies? You are learning it from your high school, and it is nothing but Drosophila is the answer. Then for studying insect spermatogenesis process, which target organ need to be focused? Spermatogenesis, production of or the formation of sperms. So you have to go for the male reproductive organ. Then next one, for generating insect vector genome data. Now genome data, which is essential and primary requirement. RNA isolation. RNA is for transcriptome studies. That is, you isolate all the RNAs present in that particular insect and you do sequencing and you study everything regarding the various RNA in the insect. So here they have asked genome data. So naturally you have to go for DNA isolation and sequencing. So the answer here is option 2. Then find the correct statement. Insect carries innate immunity but lacks adaptive immunity. Yeah, that itself is correct statement, because insects have got innate immune system but they lack adaptive immunity. Means it will include your antibody production. So insects don't produce any antibodies, so they lack adaptive immunity. So first statement is the correct statement. Then which of the chemical agents would be most suitable to isolate DNA, RNA, protein together from the same sample? It is your triazole. Triazole, it is acid guanidinium phenol based reagent, and it can be used for extraction of DNA, RNA, protein together in the sample. So what you take is, suppose you are isolating it from the leaf sample, you homogenize the sample with adding a triazole reagent. Then after that you add a chloroform and you can see the phase separation after you add the chloroform. So on top you have got the aqueous phase and it contains your RNA. 1 minute. Okay, this is the aqueous phase and it contains your RNA over here. The interphase, it is having DNA, and the organic phase, it will have your proteins. Then as per your requirement, you can purify whatever sample you want from these 3 phases. Okay. Then next one. Both adult male and female feed on blood. Here they have not given the name of the arthropod. So I am taking it as mosquito based on after referring the answer key. Okay, so I got the answer and I referred the answer key and I found that maybe the question is for mosquito. So if you take mosquito into consideration, both adult and male and female feed on blood is incorrect. Then so statement 1 is incorrect. Both adult male and female feed on nectar sugar, it is correct. Females also feed on nectar sugar, not only blood. Okay. So statement 1 is incorrect, but statement 2 is correct, makes option 4 as the right answer. Then given below are assertion and reason. Okay. The level of efficacy of inactivated viral vaccine differ for different viruses. Yes. That statement is correct. Now why? We will see the reason. Because the inactivated viral vaccines are grown in a variety of cell substrates such as eggs, monkey, kidney, cell line, human diploid fibroblast or mouse brain. Okay, so these during the production of inactivated viral vaccines, you grow them in different cell substrates. It can be either egg or monkey kidney cell line or human diploid fibroblast or mouse brain. And these viruses are inactivated with chemical or detergent. So because of this variation in different cell substrates, and you inactivate either with chemical or detergent, the efficacy of inactivated viral vaccine differ for different viruses. Okay. So both A and R are true, and R is the correct explanation of A. So option 1 is correct. Then next one, again assertion reason. The Western Hemisphere was eliminated of polio viruses by 1990 as a result of immunization program. 100% correct. Then why was this eliminated? Because of immunization programs. They have two polio viruses, oral and injected one. Okay. So introduction of two polio virus vaccine, that is inactivated polio virus vaccine IPV and oral polio vaccine to the population from 1955 to 63 led to the elimination. That is the correct reason for the assertion. So both A and R are true, and R is the correct explanation of A. Then next one, which virus undergo genetic drift and genetic shift? It is your influenza virus. Now what is this genetic drift and genetic shift? It is also called antigenic shift or antigenic drift. Now first we will see the antigenic drift. Okay. So it is a small gradual genetic change, like due to point mutations that can happen in the virus genome, and it will result in a new virus which will have the changed viral surface proteins, that is your hemagglutinin and neuraminidase, and this will result in an epidemics only to a concentrated only to a certain region. So accumulation of mutation over time, you got virus B. So see here, this is red is the mutation in the viral genome. It has resulted in different protein. Viral coat protein is different from this. Here it is triangle, here it is somewhat like a rectangle. Now see what is genetic shift or antigenic shift. So here what is happening is there are two viruses. One is virus A, virus B. We will take influenza virus A. So influenza virus type A as well as influenza virus type B, both are infecting the same host. Or I can say they are infecting the same cell. And as a result of this infection, these two viruses, they will come together and they will shuffle their genetic material, and this will result in the production of a new virus. See virus C, you got some genes from virus B, red one. You got some genes from virus A, that is the blue one. Entirely different viral protein, that is hemagglutinin and neuraminidase. Even though here it is a circle and instead of a triangle in both the cases, you got what is that, a rectangular shape protein, and the color is also entirely different from both the virus A and virus B. See here. So this kind can result in pandemics. Okay. So that is what is called antigenic shift and antigenic drift. Then next one, highly effective antiviral drugs have been developed for all viral diseases. No, not for all viral diseases. Why? Because these viruses, they have a tendency to quickly mutate, and they will develop resistance to existing antiviral drugs. That is one reason why highly effective antiviral drugs have not been developed for all viral diseases. Okay. Another reason is that some viruses are difficult to target because they will be using the host cell machinery. Suppose a type of virus is infecting human beings and it is using the human ribosomes for synthesizing its proteins. So when you target against a ribosome thinking that the virus is utilizing its own ribosome and not the host ribosome, you are going to affect the host. The viral will not, of course it will be affected, but host also will be affected along with the virus. That shouldn't be a proper drug development strategy where you are causing negative to the host. Okay, so that is one reason why highly effective antiviral drugs have not been developed for viral diseases. So statement 1 is not correct. Okay, now come to statement 2. Drugs that are very specific for viruses, drug targets are very specific for viruses. Okay, yes, that is correct, because drug development, they always target specific viral proteins. Okay, so these kind of drugs are developed only against one particular virus and not against another virus, because each virus have got different viral proteins. Okay, now suppose you take the case of that influenza virus. After mutation, you got an entirely different coat protein for influenza virus. Now the drug that you developed for the parent virus, that is the one without mutation, will not have any effect on this mutated virus because it is having a different coat protein. Okay, so drug targets are very specific for virus. Therefore, only few antiviral drugs have been developed against one virus and active against another virus. Yes, that is correct. So here what you will do is some antiviral drugs, they are developed in such a way that they target specific proteins or conserved proteins among multiple viruses within the same family. That is important. Not from different family. Within the same family. So within the same family means if you target some proteins which are conserved among the various viruses belonging to this particular family, you can develop an antiviral drug. Suppose it is you are targeting the viral polymerase. Viral polymerase, they are conserved among viruses belonging to the same family. So when you develop the antiviral drug against the viral polymerase, it is active against one virus as well as active against another virus belonging to the same family. So statement 2 is correct. Drug targets are very specific for viruses. Therefore, only few antiviral drugs have been developed against one virus and active against another virus. Okay. So statement 1 is false and statement 2 is true. So the answer is option 4. Now next one, match the viruses with their respective family. So rotavirus, it is for Reoviridae A3. Okay, A3 is there only in option 2. So happily option 2 is your answer. HIV, it is Retroviridae. If you are 100% sure and you don't know anything regarding other viral families, if you know B as Retroviridae, happily you can mark option 2 as your answer. Then influenza virus, Orthomyxoviridae. Okay, C1, and poliovirus is picornaviridae D2. So answer here is option 2. So thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question paper of ICMR being discussed, you can go to my channel Omni Bioentrance. In the channel playlist, see for ICMR, and you will find them there. Thank you.